So the topic today I'm going to discuss is the anterior-posterior axis specification in Drosophila. This topic is very important, as I said, and you can see here that the Drosophila has a head region, and this is the anterior region. Okay, this is the front part, and in the back side you can see here this is the posterior region, and if you draw an axis from this anterior to posterior, so this forms the anterior posterior axis. Okay, so this is also called as AP axis, right? So you can see this AP axis plays a very crucial role in the life of Drosophila, because the body of Drosophila consists of three parts: head, thorax and abdomen. You can see this is the head region, thorax and abdomen. All these three parts are formed along the anterior-posterior axis. That means without anterior-posterior axis there is no head, thorax and abdomen. It means Drosophila cannot really differentiate its zygote. So this anterior-posterior axis formation starts when it is in the egg state, and it continues through the zygote stage. And one thing again you should think is two structures, acron and telson. The acron is present in the anterior side and telson is present in the posterior side. So as we said, the anterior-posterior axis of Drosophila is influenced by 40 maternal genes, about 40 maternal genes. Maternal genes are those genes which are inherited from the mother. That means that comes through the egg cell. Okay, so the egg cell itself is polarized in a way of anterior-posterior axis. So these maternal genes establish some signaling centers which help in the anterior-posterior axis specification. So there are three classes of maternal genes, generally called as egg polarity genes. So we will see which are those classes. So first class of genes is called as anterior signaling center, that leads to the formation of head and thorax regions. And the second center is called as the posterior signaling center, which leads to the formation of abdomen. And finally, the third signaling center called as the terminal signaling center, which leads to the formation of acron and telson, which we mentioned a little earlier. So now we will see which are the genes involved in each of these three signaling centers. So first we will see the anterior signaling center. So the major genes are bicoid, hunchback, Stophan, exuperantia and swallow. So the bicoid and hunchback play the most crucial role, actually, where these two are known as the anterior morphogen. That means where the hunchback is expressing that region becomes the anterior side. Okay, and actually it is the bicoid gene activates the hunchback. Okay, so where hunchback is expressing, that region is becoming the anterior side. Okay, you got it. And there is another gene called caudal, and where caudal is expressing, that region becomes posterior. So both of these genes, mRNAs, are uniformly distributed along the egg. Suppose this is the egg. You can see that this throughout the anterior to posterior, both these genes are uniformly distributed. But then how this is possible to specifically control the expression of hunchback and caudal? So that is mainly, one thing is bicoid, which is located in the anterior region, that activates hunchback, and the bicoid also does one thing, that is bicoid suppresses caudal in the anterior region. So that means caudal will not be present functioning in the anterior region. Okay, so another thing is, there is another gene called nanos. Nanos actually inhibits hunchback. So nanos will inhibit hunchback. So nanos is present in the posterior side. Okay, so this is usually present in the posterior side. So we will see. So here you can see that three genes, tofen, exuperantia, and swallow, they help in the localization of the bicoid mRNA in the anterior side. That makes the bicoid to help hunchback express in the anterior, and thereby forming the anterior surface side. Okay, so then coming to the posterior signaling center, you can see that the nanos, which I just mentioned, the nanos suppresses hunchback. This is what it does. Okay, so and actually caudal doesn't need any activator. So as the bicoid activates the hunchback, the caudal doesn't need any activator. Okay, so other genes, Oscar, tofen, etc., they all help in the localization of the nanos mRNA towards the posterior region of the egg. And now finally, a terminal signaling center, which is involved in the acron and telson. The genes are torso-like trunk, etc., which all help in the formation of acron and telson. So now I'm trying to explain what I said earlier in the diagram. So with the help of this diagram, we can see that the bicoid concentration, I'll change this pen color a little bit so that it will be easier for you to understand. Okay, so the bicoid is present in the anterior region. Okay, the concentration of the highest is at the anterior-most region, and therefore it activates hunchback in the anterior region. It causes the translation of hunchback in the anterior region, and meanwhile nanos will inhibit hunchback. Okay, so the concentration of hunchback will be very less in the posterior region. So hunchback makes the expression of hunchback makes it anterior. Okay, so this is how the anterior side is formed. And you see here the bicoid, the caudal is usually highly produced throughout the caudal mRNA is present throughout the egg, but in the anterior region the bicoid inhibits the expression of caudal, or the translation of the caudal is inhibited by the bicoid, and therefore the concentration of caudal will be very less in the anterior region, thereby higher in the caudal, higher in the posterior region. Okay, so the expression of the caudal in the posterior region leads to the posterior specification. So this region becomes posterior region. Okay, so but the nanos is not activating caudal, that you should remember. Okay, so once again, bicoid activates hunchback, it suppresses caudal. Nanos doesn't activate caudal, but it suppresses hunchback. So thereby a gradient of the hunchback is established from anterior to posterior, while the hunchback gradient is highest in this and then lowest towards the posterior. Meanwhile, the caudal gradient is highest in the posterior and lowest in the anterior. So this is how the anterior-posterior axis is established in Drosophila, and this is only the beginning of this topic. Actually, we only dealt maternal genes. Now further development of Drosophila involves further many more genes, which I'll be dealing in the later sessions. So if you have any doubts, you can comment it in the comment box, and I'm doing videos in Malayalam also, so you don't have to get confused. I have kept a separate playlist for English videos, so keep watching. Thank you very much.